selections interact to lead to the evolution of traits that allow individuals to blend into their environment, as well as traits that enable individuals to stand out amidst their environmental context. And from a larger scale perspective, we've also accumulated a lot of beautiful studies about phenotype divergence among closely related groups, as well as population genetic structure. But there's still a lot of outstanding questions when it comes to processes that underlie patterns of diversification. And a particularly fruitful way of thinking about this really large question is to look directly between two scales of analysis. That is, everything we know about how phenotypes evolve within populations, and asking whether or not there's a dynamic and important interplay between those evolutionary processes and those larger scale patterns, thus allowing us to examine and predict the process and their emergent patterns. Such an approach is really only appropriate, as the speakers before me have alluded to, between closely related populations that have variation in phenotype and are geographically isolated, that are early in the process of divergence, so that we can conduct a direct analysis of how and why populations emerge. A particularly suitable study system for addressing these questions between phenotype evolution and diversification is the Barnes-Wallace species complex. This is comprised of 6 widespread subspecies shown here in this distribution map. All of these species inhabit a fairly similar ecological context across the northern hemisphere, but there's interesting variation with respect to morphological variation as well as behavioral variation in terms of migratory behavior. Another interesting thing about our system is that it's particularly tractable. We can catch hundreds of breeding individuals. We can collect a variety of morphological data sets with respect to elements of phenotype that are relevant to sexual and natural selection. We can individually mark populations and get really great fitness estimates for individuals as a function of phenotype evolution. Another amazing trick of our system is that we can manipulate relevant aspects of phenotype, and I'll be talking about tail finger length, which we can FedEx between populations and glue on to individuals from one population to the next. And we can also manipulate an individual phenotype with respect to ventral coloration. So we've conducted phenotype divergence experiments in 3 closely related populations. And when we do this, what we find is that the phenotype divergence that we observe in these populations naturally, we have now causally demonstrated that the degree of phenotype divergence is underlain to some degree by divergence of sexual selection. So this is great. So we now have one aspect of understanding the connection between phenotype evolution and this larger scale pattern of phenotype divergence in this closely related group of populations. But we're next interested in asking what all this phenotype divergence might be doing in terms of population genetic structure. So to ask that question, we sampled 8 closely related populations across 4 different subspecies with a sample size of 350 individuals. And we basically looked at 2 different analyses. The first is asking whether or not there's a simple pattern of isolation by distance in these populations such that pairwise geographic distance is somehow responsible for pairwise population genetic difference. We also wanted to test a model of isolation by adaptation, so taking all of those aspects of phenotypes that we know are divergent and asking whether or not they explain patterns of population genomic divergence. So in essence, we asked 3 questions. First, do we see a signature of isolation by distance? Next, do we see a pattern of isolation by adaptation? Do those traits that we know are underlain by divergent sexual selection play a role in the buildup of genome-wide divergence? And then can we look at these 2 models side by side to better understand whether it's geographic distance or whether there's something about adaptation that's explaining patterns of genome-wide divergence? So we conducted a large genotype and phenotype analysis comprised of those 350 individuals. We generated 100 base pair reads on an Illumina high-speed platform. We identified 26 plus hits that we aligned to the barn swallow genome. And what we found is that these populations are indeed really closely related to one another, such that the average FST between these populations is about 0.4 and ranges from 0.01 to 0.07. So first, do we find a pattern of isolation by distance? It would be surprising if we didn't. So we do find that average genome-wide divergence accumulates as populations become geographically distant to one another, but I want to draw your attention to the scale of that y-axis. These populations are really, really closely related to one another. The next thing we wanted to do is take those aspects of phenotype that we know are underlain by sexual and natural selection and ask if they're influencing genome-wide divergence as well. We also collected a data set of relevant features of the environment that are important for the aerial insectivores that our barn swallows are. Features of the environment that include precipitation, temperature patterns, as well as looking at elevational differences. So do we find any influence of phenotype divergence on average pair-wise FST between these closely related populations? And the answer is yes. The aspects of phenotype that we measured, wing length related to migratory behavior, streamer length, throat color, breast color related to sexual signaling, all explain a lot of variation in the buildup of genome-wide divergence between these closely related populations. Surprisingly, we didn't find any influence of our environmental features that we know that aerial insectivores are sensitive to. So we next wanted to conduct a variance partitioning analysis by looking at the relative influence of isolation by distance and the relative influence of isolation by adaptation. And this is a particularly nice analysis to conduct because we know that these matrices, that is, features of morphology related to natural selection, which is this wing matrix here, features of morphology that we know are underlain by divergent sexual selection, coloration matrix, as well as this matrix of pairwise geographic distances. We know that they're intercorrelated to some degree, and we wanted to be able to account for those intercorrelations in an analysis. What this analysis enables us to do is basically condition the influence of one particular matrix, accounting for those intercorrelations. And so this is very intuitive. It's a Venn diagram. It shows you that there is indeed overlapping variation among those three matrices, but that when you condition pairwise distances in wing length for correlations between coloration and geographic distance, we see that wing length, differences in wing length, is responsible for a lot of differences in genome-wide variation. Coloration is also contributing about 5% variation in explaining genomic differences among these populations. And when we partition the influence of geographic distance and its correlations with phenotype, we find that all of that variation is being consumed by these other two phenotypic matrices. A redundancy analysis confirms what this variance partitioning analysis is showing us, in essence telling us that pairwise differences in wings, pairwise differences in coloration explain an astounding amount of variation in the buildup of genome-wide divergence, controlling for that influence of isolation by distance. And so yes, we have a pattern by isolation by distance, and I've also shown you that differences in wing shape and length, as well as differences in ventral coloration, explain a lot of variation in genome-wide divergence. When we partition the variance between isolation by distance and isolation by adaptation, we find that overwhelmingly a model of isolation by adaptation trumps a model of isolation by distance. Okay, so great. So we've got the buildup of genome-wide divergence in these largely allopatric populations as a function of these phenotypes that we know are underlain by an adaptive process. So what happens when populations come together? So to conduct sampling across parts of the barn swallow species distribution where we speculated that populations would come into contact, Liz Scordato, Matt Wilkins, and Georgi Semyonov traveled from Moscow all the way to the city of Japan. What I'm showing you here in red is that continental rustica population. In purple is that North Asian Tytleri subspecies, and in blue is that South Asian Gutturale subspecies. And as you can see, as they traveled across this transect, they encountered populations that looked like intermediates. And so here's what rustica and Tytleri look like in hand in this region. And then as they continue to travel, this is what the phenotypes look like as Tytleri meets Gutturale. So the next thing we wanted to do is do some genomics and phenotype analyses on these populations across that large transect. This is a data set comprised of 534 individuals, 3 subspecies, 2 contact zones that I've just shown you. We use the same genotype sequencing approach as we did before. We aligned more than 20,000 SNPs to the draft assembly of our barn swallow genome. And I'm going to show you the results of a Bayesian admixture proportion model to identify the ancestry of individuals across the transect. And so this frequency distribution shows you the proportion of admixture. On one end, we have pure parental rusticas, shown in red as you sort of go across the transect. And on the other end, we have this North Asian subspecies Tytleri. And as you can see, there's lots of parentals along this transect, not a lot of evidence of F1 individuals, but there is some evidence that we do have F2 background. So we can infer that there's fairly strong reproductive isolation going on in this particular context. Well, what about the next one? Again, I'm showing you the frequency distribution proportion of admixture, and we can see that there's a lot more evidence of F1 hybrids here, suggesting weaker reproductive isolation relative to that other context. So what about phenotype and genotype? What's going on? I've already told you that coloration is responsible for 5% of genome-wide divergence in those allopatric populations. Are they playing a role in reproductive isolation when these individuals are in contact? On this x-axis here, I'm showing you ventral coloration. Individuals on this end of the distribution are darker, on this end they're lighter. This North Asian subspecies is shown here. Rustica is shown below. And what I am showing you here is that there's just a thin band of phenotype overlap, suggesting that coloration is pretty important in reproductive isolation in this zone. On the other end of that contact zone, where Tytleri meets Gutturale, we see a lot more overlap in phenotype, but coloration still seems to be responsible for reproductive isolation to some degree. So ventral coloration predicts genome-wide divergence when populations are geographically isolated, and to some degree it seems to be really important in predicting admixture or the degree of hybridization when these populations come into contact. But if you want to learn more about what's happening in these contact zones, you need to see Liz Gordano's talk in the Spotlight session tomorrow. And also to learn more about the demographic history of these populations, you need to go to Kristen's talk on Monday. The next steps are to do some more population-level experiments within these contact zones to actually look at how behavioral interactions are mediating reproductive isolation. And then the hope is to actually look at these processes of mate selection and migration to look at the interactions of phenotype evolution and species diversity when these populations are in contact with one another. I want to finalize by thanking the various funding agencies and the many